Hi, everyone. I'm Srividya Swaminathan. I'm an assistant professor in the Department of Systems Biology at City of Hope Comprehensive Cancer Center. I'm a fairly new faculty. I joined in 2019. And briefly, I would like to describe what my lab works on. So my lab is interested in identifying cell intrinsic and cell extrinsic, mainly immune surveillance processes that are regulated in high-risk B and T cell acute lymphoblastic leukemia. Our long-term goal is to translate the knowledge gained from this to identify novel immunotherapeutic approaches and also novel diagnostic tools for combating high-risk acute lymphoblastic leukemia. So more recently, my team has been profiling the immune microenvironment in both transgenic mouse models, which enable us to study primary leukemogenesis, as well as in high-risk ALL patient samples using high-dimensional cytometry. And using this, we would be able to figure out how the immune microenvironment is remodeled and identify immune subsets that need to be modulated and can be developed as both diagnostic and for therapeutic purposes. So in one such profiling experiment, we found that the immune subset, the cytotoxic immune subset known as the natural killer cells, these are specifically modulated during the process of leukemogenesis. And the natural killer subset is suppressed in two ways in high-risk leukemia. One is the reduction in numbers of natural killer cells, and the second is the functionality of the residual natural killer cells in the ALL microenvironment is highly suppressed. By functionality, I mean the reduction in the lysis of target cells, that is the leukemia cells. So my lab is investigating the molecular mechanisms by which NK cells are reduced in numbers and in cytotoxic potential. And by doing multidimensional cytometry, we recently identified that surprisingly, the NK cells in leukemia patients are highly activated. They have a highly activated phenotype, even though they have a poor lytic potential. So patients with more activated NK cells have significantly worse clinical prognosis than patients who have lower frequencies of these activated cytokine-producing NK cells. And it acts as an independent predictor of clinical outcome, independent of normal features of worse prognosis in leukemia, which include minimal residual disease positivity and central nervous system involvement. So we have identified, surprisingly, that activated NK cells predict poor clinical prognosis in ALL patients. And this information is extremely useful because in the future, we envision that this can be developed as a diagnostic tool whereby you can conduct profiling of NK cells by cytometry using blood samples derived from the leukemia patients at the time of diagnosis to define their NK population and decide whether these patients are likely to recur in the future or not, and whether these patients will need to be administered NK cell-based therapies for sustained leukemia regression. The second clinical relevance of our project is that the knowledge of understanding how NK cells are suppressed in leukemia patients can be directly translated to engineer NK cells as NK cell-based therapies and to reverse this process of NK suppression in leukemia patients. And this mode of NK cell-based therapies has several advantages in leukemia. One is the ability of NK cells to be developed as off-the-shelf allogenic therapies, whereby they can be derived from healthy donors rather than from the same patient. And this is particularly important in treating infant leukemia, where obtaining sufficient blood from leukemia patients for engineering other cell-based therapies like CAR T cells may be clinically challenging. The second advantage is the ability for sustained regression by NK cells in cases of leukemia, which actually relapse after treatment to CAR T cell-based therapies or after treatment with induction therapies, so on. So by delineating the defects in NK surveillance, we envision that our knowledge of knowing how activated NK cells predict poor clinical prognosis can be directly translated to ALL diagnostics and treatment.